I will. I'm very happy to have the honor to host virtually. I wish he was here in person. Professor Adriano Aguzzi, who needs no introduction anywhere, especially in Israel, where he's very well known. And as you probably know, Adriano is Italian, but he's been working in Zurich for many years. He's the director of the Institute of Neuropathology at the university there, and he won all the prizes there is, pretty much, numerous prizes, including the Ernest Jung Prize, the Robert Koch Award, the Tembo Gold Medal, Marcel Benoit Swiss Science Prize, and many others, 2 advanced ERCs, everything that you want. It's really an amazing career, and Adriano has made numerous important contributions. Mostly is recognized as one of the world's leaders in the field of prions. And as you probably all know, whether you are a neurobiologist or not, everyone knows that prions are not only devastating diseases, but they also have a fascinating biology that contradicts many dogmas and that we only partially understand and keep surprising us. And if we are worried about the pandemic that we are experiencing now, if there will be a pandemic of prions, it will be a lot worse because this is much less familiar and understood. So we need to be ready for something like that as well. But of course, there's also endogenous prions that are not spread like the mad cow and so on. So this is really a fascinating field, and I'm sure you are as anxious as I am to hear Adriano present his talk that is titled "Shifts and Drifts in Prion Science." So thank you very much, Adriano.
Well, I have to tell you that it's a great pleasure. It's amazing. As Oded has said, I have often traveled to your country, and it has always been an incredible feast of science. And last year I did a mini sabbatical at Technion, which was just for a weekend, but it was really an incredible experience for me. And in a way, one of the perhaps saddest thing about the corona epidemic is that the international exchange of scientists is really what drives our entire enterprise. I think that we get ideas, we get motivation, we get enthusiasm out of talking with each other, and that's such an integral part of what we do that we cannot do without. And in fact, I would even say that science has discovered globalization and has done away with country borders long before the economy discovered that. And so now being deprived of that is, for me, I think is one of the most painful things about the epidemic. And but nevertheless, it's great to be with you. And so this time I came to Israel using Zoom Airlines, but that is good. And so hello everybody. As Oded has been saying, I have been totally monomanic about prions now for 30 years. It's pretty much the only thing I did. There is a little history behind that. I used to be a postdoc at Institute of Molecular Pathology in Vienna, and at that time, things were not going very well. And then my supervisor really told me, look, you have to focus on one thing. You cannot be spread all over. If you want to do an impact, you really have to go deep onto one thing. And I took this very much to heart. And then for the following 30 years, I worked on one single protein, which was perhaps a bit too much of a good thing. Yeah, but that's essentially my life history. So, okay, let me just share the screen, see how well this works. And right. And so this will stop other screen sharing. So let me try to see. All right. You see a big blue screen?
Nope. Now I still see you, Adriano.
So that may not work very well. So let me try to share it again. You have it now?
Now.
Yes.
Yeah.
Right. So, so first thing, just do whatever you see fit about what I'm telling you. Nothing is secret. Nothing is confidential. Broadcasted, contradicted. That's the whole purpose of this exercise. So the first story that I would like to share with you is the result of some research, and this is actually not really directly Prion's. It's a result of some research that was performed by two very talented people, Daniel Kirschenbaum, who is currently a postdoc at with Ido Admit, and Francesca Catto, who is a PhD student. She's still with me. And their idea was really to try to devise ways to visualize protein aggregates, prions or other aggregates, amyloid plaques in the brain in a holistic manner. So to actually record every single aggregate that one could see in the brain. And the reason is that the way we are looking at protein aggregates in tissue is by two-dimensional histology, and there are many limitations to that. So we wanted to have something that would also be amenable to computational pipelines. And of course, the idea was to use tissue clarification, and this is really the technology that was pioneered several years ago by Karl Dijsselroth, and who invented a technology that was called Clarity. Now, the problem with Clarity is that if you, it turns out it was, I mean, it was the start of this things, but it turns out that it doesn't work very well. And many people who have tried Clarity eventually have given up. And Daniel Kirschenbaum's idea was that maybe the reason is because the electric resistance of the brain is higher than that of the buffer, and therefore the electrophoresis by which you clarify the tissue might not work very well. And so he came up with an idea which, you know, in a way you could argue it's trivial, it's the egg of Columbus, but it turned out to work very well. And what he did was essentially to create an insulating layer between the anode and the cathode, such consisting of mineral oil, such that it would separate the positive and the negative circulation of the buffer. And this way it would force the electron beam to actually go through the tissue and remove the lipids. The whole principle of all this is that if you remove the lipids, then you end up with a polyacrylamide scaffold that is an hydrogel that substitutes essentially the cell membranes, and this is transparent. The lipids scatter the light, and that is the reason why the tissue is not transparent. So that turned out to work very well, and it's actually very fast. And in fact, this is an example of a brain that was clarified using this method that we call crystal in just a few hours, and you see that you can see through it. There is a millimeter paper underneath this mouse brain, and then the question was how can we actually visualize protein aggregates? Problem is these methodologies are very hard to use in conjunction with antibodies, and one reason is because antibodies are large molecules, and the pores that are created during this tissue transparentization are simply not large enough. And but also antibodies are obviously sweeter ions that are not charged at physiological pH. You can shift the pH, but then at some point you will denature the antibodies and you will impair the binding to the antigen. So we decided that we would use something else, and what we decided to use is highly charged small molecules, and I will explain to you what it is. But the intuition of Daniel was to confine these dyes to a small capsule of agarose, and then again to use electrophoresis to drive the dyes through the tissue and therefore to stain the protein aggregates. So the whole idea was to create something that would look a bit like an espresso capsule that you would insert into a little machine that with 3D printed, and then we could actually stain aggregates. Now, as far as the dyes go, what we decided to use are polythiophene, and these are compounds that were described by a former postdoc of mine, Peter Nilsson, who is an organic chemist by training. And Peter had this idea that the thiophene may actually intercalate with amyloid. And as you see, these are called also luminescent conjugated polymers. And conjugated means in chemistry that you have two bonds followed by one single bond, followed by a double bond, and again a single bond. And of course, this creates a resonant pi electron system that actually determines the fluorescence of these compounds. And the nice thing about this is that there is rotational freedom. The single thiophene monomers can rotate against each other along this single bond, and they can assume, for example, a twisted orthogonal conformation or a planar conformation. And the amazing thing is that the emission spectrum completely changes. And this is the same polythiophenic acetic acid at 3 different pHs, and in polythiophenic acetic acid means that you have a COOH residue here, which of course will attract or repel depending on its protonation, and that is why you get all these different colors. Now, this was pretty nice, but then we thought, okay, maybe we can actually show that this intercalates with amyloids, because the way prions replicate is that you have monomers that will assemble into large highly regular, highly geometric aggregates, and the distance between the beta sheet in these aggregates is around 10 to 11 angstrom, and therefore, the distance between the thiophene rings are around 5 angstrom. So the hope was that maybe this will just simply exactly fit into the amyloid fold and stain, and because you know, there is a lot of discussion about what are prion strains, and our idea was that maybe the strains is really different quaternary conformation of these aggregates, and because the thiophenes will fluoresce at different wavelengths depending on how they are locked into the amyloid, this may be a way of actually diagnosing prion strains, and it turns out that this works very well. So bottom line, these thiophenes, together with Beat Meyer, we did some structural studies so now we know exactly how these thiophenes intercalate. This is a prion molecule, it's a HETS, it's a yeast prion, and you see that the thiophenes go into this hydrophobic groove, and then the COH, the carboxy residues, they interact with lysines on the surface of the molecule, and this is the little machine that we have devised for doing the electrophoresis. And then the question is, how do you, so the staining works very well, and how do you record this? And so we thought, okay, we do it by SPIM, by light sheet microscopy, which you probably know. The way this works is that you have a light sheet that is perpendicular to the acquisition pathway. So the light sheet comes, so there is a galvo, which is a rapidly vibrating mirror that will create a sheet, and this sheet will be focused in such a way that it will cut an optical section through your specimen, and then the objective will record this entire plane. And then you move your specimen across, and this way you get a stack of images. But there was no real no real machine that would do that when we started this project. So what we decided to do was actually to build such a machine, and this was the work of Daniel Kirschenbaum together with Fabian Vogt, which was published in Nature Methods last year. And this is essentially the thing. I know it doesn't look like a microscope, but it does work, and you see the guys have helpfully put in all the labels. So you see here is actually the camera, and here is actually the specimen. Then here is a chair, just in case. Yeah, I know they will try. They were trying to make it fun. And then we bought some lasers. And because the guys like a lot of colors, then they asked me to buy more lasers so that we could even get more colors, and then even more lasers. And then actually, when they when they built the machine, they made a time-lapse video so that you can exactly see how this is done. And in case you want to build this in your home, you can use this from YouTube, and you can use it as a blueprint. So this is how you make selective plane illumination microscope. And right, at some point I see that Fabian is lying on the optical table. That may be a somewhat unorthodox way of doing things, but eventually this is the kind of images you can get here. So this is now a transgenic mouse that expresses the amyloid beta protein of Alzheimer's disease, and these are plaques. The yellow guys are Alzheimer plaques, and at the same time it expresses GFP under the control of the Claudin 5 promoter, which is expressed in brain endothelia. And these are mice that were made by Christer Betzold at Karolinska. So here we have a simultaneous visualization of vessels and plaques. And yeah, I couldn't resist quoting Oded. So what is the physiological relevance of all this? And this is a little movie that was made by Imaris that actually visualizes the entire vascular tree. So we have everything here. We have the arteries, the veins, the capillaries, everything, as well as the plaques. So the little critters, the little spheres that you see in the middle are actually the Alzheimer plaques. And then, you know, once you have this as a data set, as a image stack, then you can start doing interesting things, no? Because you can start asking, for example, what is the relationship between a plaque and the next, the closest vessels, and then you can also ask questions related to therapy, because therapeutics will typically reach the brain through the vascular system. So it is thinkable that the plaques that are closest to the capillaries may be hit best. And of course, what are the therapeutics for Alzheimer's disease? At the moment, there are BACE inhibitors, which are small molecules, 200 Dalton or in this region, but there are also antibodies, which are 1,000 times larger. So you would expect that maybe the plaques that are hit by the different antibodies may be different. So the question arises whether the treatments of Alzheimer's disease may be region specific. And therefore, the guys in the lab did tried three different things. They tried a BACE inhibitor, they tried an anti-A beta antibody, which was donated by Novartis, and also an LCP, polythiophene, because we had already shown that polythiophene are active against prions. So the question was, maybe they're active against Alzheimer's as well. And then all the mice were treated for 3 months. Then the brains were clarified the way I showed it. Then the selective plane illumination microscopy was performed. And then the 3D images were registered to a brain atlas. That is, everything was normalized or morphed into a standard idealized mouse brain in order to be able to compare the voxels. And this is the kind of images that you end up with. So you have a control mouse, which will develop plaques. Then you treat the mouse with whatever compounds, BACE inhibitors or whatever. And then you end up with a probability map. Now, I really need you to understand this. What we are seeing here are not plaques. What the color codes here is the p-value for each voxel that indicates whether the therapy was effective at that particular space. And and this you can construct for treatment 1, for treatment 2, and then you can compare. You can compare whether one treatment will hit plaques in the same spatial arrangement as another treatment. And that we thought could actually be powerful. And these are coronal sections that are derived from these 3D stacks. And now the results were, in my opinion, super exciting. And really, I mean, I have to say, this completely knocked my socks off, because I was not expecting this. If you actually look at the probability map for a BACE inhibitor and that for the polythiophene, they're almost mirroring each other. The BACE inhibitors are most effective in the basolateral part of the cortex and in some parts of the basal ganglia, and the polythiophene is almost exclusively effective in the dorsal cortex. So these are things that were never appreciated demonstrated before, but it turns out that each one of these compounds have a very selective regiospecificity. And so that is where we stand with that. But then we thought, okay, now we can actually go into mechanistics. And Oded would ask probably, what is the mechanism? And so what is the mechanism? I don't know, but I thought that maybe we can start looking at atlases of spatial transcriptomics. And you know, there are such resources where you can look at which gene is expressed where in the brain. And then we can start comparing and asking whether the efficacy of therapy somehow correlates on a voxel by voxel basis with the expression of specific genes. And we can run this test for every gene that is expressed in the brain. And this was done by our collaborators in Stanford, Jin-Hyun Lee, who is a fantastic computational biologist, neuroscientist. She's an electrical engineering by training, and her PhD student, Ehsan Dalgani. And they did this amazing work, and also the computational power that was needed was remarkable. And so they compared the voxel by voxel expression of thousands of genes, and then they made correlations. And it turns out that the highest correlation, for example, to the activity of the base inhibitor is Thy1. Now, why is that the case? Well, it turns out that the APP transgene that is expressed in this mouse model is driven by a Thy1 promoter. So in a way, you could argue that this is an artifact, but in fact, it's a fantastic positive control, because of course, you get the therapeutic efficacy in those places where the transgene is expressed. So this somehow validates the whole thing. The second thing is that base itself, the beta secretase that is responsible for making the A beta peptide, again, is a very high likelihood hit. And again, it's, of course, this is a base inhibitor, so it will be active where base is expressed. But so these were kind of our sanity checks for the system. But then when we are can look actually at everything. So here we're plotting the correlation of various genes expressed in the brain against the efficacy of the polythiophene on a voxel by voxel basis, or against the efficacy of the base inhibitor. And you see that, I mean, it's very interesting. It turns out that for the base inhibitors, we find a bunch of myelin genes that are correlating. So for example, proteolipid protein is expressed in oligodendrocytes. Myelin basic protein is expressed in oligodendrocytes. Instead, for the polythiophenes, we see neuregulin, we see SNAP25, so CAM kinase. So here we see a lot of neuronal genes. So this is a part of where this stand. This is a methods development study that I wanted to share with you, because I think this is generic. It's something that we are doing for the project that I have explained to you, but it actually can be used for many other things. And of course, if any of you is interested or has a specific question that you want to ask using this methodology, we're very happy to help. And Daniel is now at the Weizmann, so he can also help you directly on site. And so the next topic that I want to quickly share with you is really what I think where all this trip is going with the question related to what are you going to do with prions. And I think that the question that is the most important one at this point is really, why are prions toxic? And I'm saying this because for the past 20 years, the focus of the entire field has been on what is the basis for prion replication. And I think this is more or less resolved. I think with that pretty much Yeah, I think there is a consensus that prions are aggregates, and essentially what happens is that you have a big fibril, and then this fibril will break into two, and that's how you get to two prions. So the fracturing of the aggregates is really what drives prion replication. It's the essence of it. But then the question that that is still open is, why is this stuff toxic? Because stoichiometrically, it's not trivial to, stoichiometrically, if you look at the amount of prions that are present even in the brain of a victim of Creutzfeldt-Jakob disease with terminal disease, is very little. So so my conclusion from this is that prions are intrinsically toxic in a very high way, and and then of course raises the question whether there are receptors, whether there are specific pathways, and whether these pathways can be whether they are actionable, whether they are druggable. And this is work that actually started many years ago in my lab, and I should say that the first hint was provided by my first postdoc when I just started my lab. That was Sebastian Brandner 25 years ago, and Sebastian did a simple experiment where he took neuroectodermal cells from embryos, from PRP knockout embryos, from PRP overexpressing embryos, and he transplanted them into the brain of a PRP knockout mouse. So you end up with a graft that overexpresses the prion protein, surrounded by an entire organism that is deficient in the prion protein. And what Sebastian showed was that if he infects these mice, he will get a spongiform encephalopathy, which is a prion disease. So you see all these huge vacuoles. Now, the huge vacuoles, this is the landmark. This is really the diagnostic feature. When you see these vacuoles, you know that you're dealing with a prion disease. And but what he saw is that only those neurons that express the normal prion protein on their surface actually experience vacuolation. Everything else outside of the graft is completely healthy, although the pathological prion protein exits the graft and distributes in the entire brain. So these already taught us that somehow the normal prion protein appears to be something like a receptor for the pathological prion protein. And then a second line of evidence came about when Frank Heppner, who was another postdoc in my lab, and again, I'm talking about things that were happening 20 years ago, but we got very interested in using antibodies to block prions, and at that time it was very difficult to make antibodies against the prion protein because because there is a strong tolerance. So what Frank Heppner did was to make antibodies using PRP knockout mice, and then he made a transgene out of it. So he made a mouse that was expressing transgenically an anti-prion antibody, and he showed that actually this mouse was protective on prions, and this was really what started then the entire frenzy of anti-Alzheimer therapy. And so it was actually the first demonstration that you can actually do something against a neurodegenerative disease using antibodies. So we made a lot of antibodies, but then I should say that, yes, some antibodies are highly neuroprotective, and this was very exciting. But then we discovered that other antibodies are ultra toxic, and these antibodies are also they also hit the normal prion protein. And this is a cerebellar organotypic slice culture. So we culture these cerebellar slices. They are 0.5 millimeter in thickness, and we can culture them for months. And what you see here is the cerebellar granule cells. Here is the molecular layer. These big guys here are the so-called Purkinje cells of the cerebellum. And now if you give this antibody, which is called POM1, it was made at that time by Magda Polymenidou, who's now a very famous Alzheimer ALS researcher, but she was at the time a PhD student. And what you see is that when you give POM1, you can destroy completely the slice. So what's going on? This antibody is ultra toxic. Now, when Tiziana Sonati showed me these results, I told powder, look, I mean, you have discovered complement-mediated lysis. This is amazing. And but it turns out that it was not, because you can also make a single chain. You can also make a single chain version that has no complement binding site, and it will do the same thing. And this is actually the structure. This is derived from the crystal structure that was of the complex of POM1 antibody with the prion protein, and this is the contact surface, and the contact surface is around 500 square angstroms. It's like a button. It's really, you push the button using this antibody, and you drive the neuron into apoptosis. Now that was ultra interesting, and then there were other things that we found that were ultra exciting. But the bottom line is, and this is my interpretation of all this: these antibodies are prion mimetics. They're prion mimetics, and they elicit exactly the same signal transduction pathways that you see in bona fide prion infections. Now this is to me, is a major breakthrough, because it now allows us to do interference screens, not having to rely on infectivity, because the antibody does not generate infectivity. So we thought, okay, now we can do perhaps some smart things. And and the first question was, okay, what is it really that this antibody is doing? I mean, maybe, you know, maybe it induces a conformational change in PRP, which then triggers the whole thing. But, you know, Michael James, who is a fantastic crystallographer, he told me, look, I cannot see anything happening to PRP. And so this was kind of a bit of a bummer. But then I met Luca Varani, who is an NMR guy in Bellinzona, and Luca was telling me, you know, you should try molecular dynamic simulations, because the crystal structure will not tell you the entire story. And from that, what Luca did, he found that there is this loop here, this unstructured loop within the prion protein. This is the binding site. This is a POM1 binding site, the red area in the previous movie. And he was saying, well, you know, this loop is very flexible, it's very floppy, and in fact, it's so floppy that you can barely see in the NMR. And the reason is because there is quite a distance between this histidine and this arginine, and they don't interact essentially. But when POM1, when the toxic antibody comes in, then what happens is that there is a whole network of hydrogen bonds that arises here. This is the antibody, this is the prion protein, and this is essentially the molecular basis for the binding site, the epitope-paratope interaction. And one consequence is that the side change of this arginine comes very close to the histidine, and it locks it. And then the question was, okay, is that really the reason for the toxicity? Because if that's the case, maybe that's how prions damage the brain. And so what Karl Fronzek did was to create a bunch of mutants. And essentially what he did was to mutagenize every single antibody into, every single residue on the antibody into alanine, in order to probe which parts of the epitope binding would be important for toxicity. And the hope was to get something that would retain the affinity for the PRP, but would no longer create this latch. We call it the H latch. And here are all the mutants that he made. You see, this is LC means light chains, HC means heavy chains. So this is the heavy and the light chain of the antibody. So he mutated all these residues into alanine, and he measured the affinity. And this is the affinity measured by surface plasmon resonance, and this is the affinity measured by fluorescence resonance energy transfer. And you see that the wild type antibody has the highest affinity. Some of the antibodies lose affinity completely. And this is not surprising, because of course, at some point, you know, these interactions are actually what keeps the antibody together with the antigen. So if you take them away, it's like if you take a leg off from a stool. It will, but you see that actually there is a number of antibodies that retain the affinity, but this antibody no longer creates H-latch. And now I want to focus on this one, tyrosine 104 mutagenized into alanine, so Y104A. So this antibody retains the affinity. It does not make the H-latch. It's unable to lock the flexible loop into place. And now this is an experiment where we take again these organotypic brain slices, and they are from the cerebellum. So you see here the beautiful neuronal contour of the cerebellum. This is the so-called arbor vitae of the cerebellum. And you see that if you infect them with prions, this is non-infectious brain homogenate. These are prions, and you see that the prion infection within 5 weeks completely wipes out the entire cerebellum. However, if you keep a single chain FV version of the mutagenized POM1 antibody, you can prevent the prion-induced neurodegeneration. And for me, this is a very satisfying result, because what it means is that the mutagenized antibody has now engaged the normal prion protein, is not toxic because it does not create this latch, but now it prevents the pathological protein to come in. And I mean, that is the interpretation. There is no direct evidence for that, but I think this is the most plausible scenario, is that the mutagenized antibody is now acting as a dominant negative mutant that will prevent, that will still bind, but will now prevent the toxicity of the prion. Right, and now, so that is one story related to the toxicity. And now, in the last 5 minutes, I want to tell you a totally new story that this is completely due to amazingly talented senior scientist in my lab. His name is Ashwin Lakkaraju, and he's a great cell biologist. He's really has incredible intuition. And when Ashwin first came to my lab as a postdoc, he asked me, so what do you think is an exciting thing that I could do? And I told him, look, you know, there is one thing that I've been wondering for 30 years about, and I have no idea how it comes about, but I think it's important, and that is the vacuolation. You know, all these diseases, they are also called spongiform encephalopathy, and the reason is because of spongiosis. Spongiosis means that it's like a sponge, and it is like a sponge, because you see how the brain looks like. This is from a human brain, is a victim of Creutzfeldt-Jakob disease, and what you see here is that are all these vacuoles that arise in the brain, and you know, this is so typical, and it appears so consistently in every single prion disease of human and of animals that I always thought that this cannot just be an epiphenomenon. It is telling something about the nature of the disease, but I don't know what. And so I told Ashwin, so Ashwin, why don't you find out how spongiosis comes about? And I think he solved it. And I want to show you the evidence. So what are these holes? These vacuoles arise inside the neurons, and particularly inside the processes. Here you see a myelinated process, and then all of a sudden it blows up and creates a vacuole. See, there is all the myelin around, so you see that the vacuoles are not between the cells, they are inside the cells. Now, how do they come about? So this is Ashwin, and these are the questions that he has asked. And the first thing he came about was a candidate approach. He just read the literature, and he said, look, you know, there is this molecule, it's phosphoinositol kinase, it's called PIK5, and it's a kinase, it's a lipid kinase. So it takes phosphoinositol 3-phosphate and it adds a phosphate residue, and the result is diphosphoinositol diphosphate 3,5-diphosphate. And what is known about PIK5 is that it regulates the size of the endosome. And in fact, if you lose PIK5, the endosome blow up and you end up with big vacuoles. was. So the question that has been asked was, could it be that PIK5 plays any role in the creation of the spongiosis? Now, the other thing that you need to know is that PIK5 exists as a complex with several proteins, but an important protein is called PIK4, and PIK4 is a phosphatase, and PIK4 can dephosphorylate the diphosphate and create the monophosphate inositol again. But, and this is new, PIK4 can also dephosphorylate PIK5, and it can control the activity of PIK5. And then there is a scaffolding protein that is called RPIK5 or VAC14. Now, this is very recent, is the cryo-EM structure, and it's beautiful, and it turns out it's very surprising. It turns out that VAC14, the scaffolding protein, exists as a pentamer, but it's very strange. The pentamer has only one binding site for PIK5. So for 5 molecules of VAC14, you have only 1 molecule of PIK5 and 1 molecule of PIK4. And as I said, PIK4 can dephosphorylate PIK5. Now, this is what happens if you put in shRNA against PIK5. You see that this is LAMP1, so you're looking at late endosomes, lysosomes, and you see that they're nice and small in the control shRNA. You knock down PIK5, they start to blow up. No? So you get kind of a microvacuolation of the cells. Now, then the question is what happens in prion infection, and what happens is amazing. I mean, this is beyond what I could have expected. I mean, this is these are mice that have been inoculated with non-infectious brain homogenate, another mouse non-infectious brain homogenate, 2 mice inoculated with prions. Here is PIK5. You inoculate with prions, the mice are sick with prions, PIK5 disappears. But only PIK5, because the other 2 components of the complex, PIK4 and VAC14, they're still there. So the prion infection leads to selective depletion of PIK5. Now, what happens to humans? These are all brains of people who died of Creutzfeldt-Jakob disease. And you see, again, you have a PIK5 here in the controls, and it is reduced. It's also very interesting. It turns out there is type 1 and type 2 CJD. I won't go into the details, but it's particularly in the type 2 form of Creutzfeldt-Jakob that you get a depletion of PIK5. Now, the important thing is that all of this is post-transcriptional because the RNA is stable. You don't, the RNA level, I'm not going to show you the data, but the RNA level are completely unchanged. So it's something that happens post-transcription. Now, so how do you study this? Well, you can't do it in mice. You need a cellular model. So we started to investigate GT17 cells because they are a nice model for vacuolation. If you infect them with prions, here again, RML means Rocky Mountain Laboratory. Prion is a strain of prions. And so you see that, again, we use always non-infectious brain homogenate. And you see, this is just a succinimidyl ester is just a generic protein stain, so you see all the proteins here, and what you see is that if you infect the cells with prion, you see the vacuolation coming up, and again PIK5 also in the cells disappears. So this is not so now we have a cellular model. We can actually study what happens to PIK5. So the other super exciting thing, I told you that POM1 is a prion mimetic antibody. So the question was, what happens to PIK5 if you give POM1 to the cells? Well, these are cerebellar organotypic brain slices, same system I showed before, treated with control immunoglobulins, POM1, you see again, PIK5 disappears. This is a quantitation, and these are the other components of the complex and the loading control. So also even the antibody quenches PIK5. Now, how does this work? Well, some work that was done by Giovanna Mallucci almost 10 years ago showed that ER stress is really important for the pathogenesis of prion diseases, and particularly the PERK pathway of ER stress, which leads to the phosphorylation of EIF2 alpha and to inhibition of protein synthesis. So then we asked whether this is what creates, what screws up PIK5, and indeed, what we see is that the activation, the phosphorylation of EI to EIF2 alpha precedes the degradation of PIK5. Now that is obviously a bit, you know, it doesn't prove anything, but more importantly, if you inhibit PERK, and with this compound, then you can actually rescue PIK5. And this is the quantitation of this Western blot. So you see that this is PIK5 with normal brain homogenate. You give prions, PIK5 goes down. But if you prevent the PERK arm of ER stress, then you can partially rescue the PIK5. So this seems already to play a role. And now the question is, why is really PIK5 gone? And Ashwin loves to do metabolic labeling. This is his passion. So, you know, every time if I let him do something on his own for a while, he will immediately do a pulse chase experiment, and he does this very well. And what he shows is that in the presence of ER stress, what happens is that the half-life of PIK5 measured by pulse chase actually decreases. And so that is already quite interesting. And the question is, could it be that the PIK5 undergoes some post-translational modification? And again, Ashwin had this amazing intuition. He thought, well, you know, this thing sits on the cytosolic aspect of the endosomal membrane. So maybe it's palmitoylated, because this is something that, you know, if it is localized to a membrane, maybe it has some kind of lipid residue. So he then established the technology of acyl rack, and I won't go into the detail of this, but this is a way of demonstrating the palmitylation of proteins, and he indeed discovered that PIK5 is palmitoylated. Now, there is a bunch of enzymes called the DHHCs, which create palmitylation these are the acyl transferases, and so it turns out. So in another experiment, he showed that indeed what happens in prion infection is that the acylation of PIK5 is impaired. Now this is super exciting. So now we are starting really to see the entire biochemical pathways of what drives vacuolation, and the conclusion here is that it is really the reduction in acylation may be what is delocalizing PIK5 from the endosome and rendering it susceptible to degradation. So there are 23 acyl transferases in the human genome. So then as we knock down each one of them in order to find which one is responsible for acylating PIK5, and he found there is two of them, DHH9 and DHH21. And if he combines the knockdown of both, then you essentially remove all acylation of PIK5 and you get vacuoles. So simply removing the acyl transferase suffices to induce the vacuolation. And then we found which are the residues at which PIK5 is vacuolated. I won't go into the details, and I think that I will try to now to come to a close, because I see that I am already overstaying my welcome. But let me just tell you one bit of ultra exciting result. So I mean, my thinking here was that well, you know, if eventually all that it boils down to is that PIK5 is making the 3,5 the phosphinosytol diphosphate, maybe what about we just give the phosphinosytol diphosphate to prion-infected cells, and we ask whether we can cure them from prion diseases. So we had a company synthesize water-soluble version, a body pi conjugate of phosphinosytol 3,5-2-phosphate, and we gave it to cells. And it's amazing. I mean, I tell you, this is incredible. I mean, these are the vacuoles. These are non-infected cells, no vacuoles, RML 6 prions, lots of vacuoles. We give what I call the BPIP, the body pi, phosphinosytol phosphate, vacuoles go down. Not only that, but also the lysosomal enzymes, which are heavily deregulated after prion infection, they also get normalized by the BPIP. And the most amazing thing is really that when you infect brain slices with prions, you destroy them, but in the presence of BPIP, the morphology of the cerebellum in prion infected slices is retained. So yes, and the same happens actually to POM1. Also, POM1 kills completely the slices if POM1 is pre-blocked with a prion protein, so that it will no longer be able to bind to the surface of the neurons, then it will not be toxic. But if you give the BPIP, again, you can rescue, at least partially, you can rescue the morphology of the arbor vitae of the cerebellum. So I think that this is the model of how everything works, but I won't go into the detail. I just leave it at that. And I want to thank all my co-workers. I think that they are amazing. They have done really an incredible piece of work, and it's very much of a teamwork. As you see, I mean, in the papers that we publish, we always have 3 or sometimes even 4 first authors, because we are big believers in teamwork and in getting stuff done together, together being more than the sum of the parts. Many thanks for listening to me.
Adriano, that was a, can you hear me? Yeah. That was an amazing talk. So many things. Yeah. Before I give the audience a chance to ask questions, I want to take the advantage of the fact that I have the microphone on and ask you 2 quick questions. One of them regarding the method, Crystal. We have, normally, you know, we work with mice, where we have a project where we work with collaborators in Arizona and have an amazing brain phenotype. Can we take these mice and make their brains transplant, or do we need to make something in advance before we start the experiment? Because
yes, you need to perfuse them with a special buffer, and we'll give you the recipe of the buffer. It's very easy. And then once you have done that, you can just send us the brain and we'll do everything. And you will get approximately 100 terabytes of data back that show what happens inside the brain.
Amazing. So I will speak with you about it. It's really exciting. And the second question is about the acetylation in the end. There are so many acetyltransferases and so on. So it's like a code, I imagine. And could this, and if Prion works on this pathway, could this be part of, and I'm speaking out of total ignorance because I know nothing about this, but could this be part of the species barrier or the species differences, these types of subtle differences in modifications of the protein?
Absolutely, and I'm not sure whether this is really relevant to species, but something that I really think is important is the cellular specificity, because prions exhibit something that we call selective neuronal vulnerability. There are very specific areas that are hit by prions, and others are left pretty much alone. And the question is really here, why is that? And the metabolism of phosphinoinositol may be there may be really a code there, and so I think you're spot on.
Thank you. And so, okay, if anyone wants to ask questions, please just turn on your microphone and ask, and we'll try to have order in the request. Yes.
Pablo, can I go for a question? Yeah. And thanks again for the fantastic talk. And I'm puzzled really about the beauty of the the data that you show about the vasculature and the plaques. And I was wondering if you guys have tried to dive into where exactly in the vasculature compartments do you see an increased deposition, not just in the cortex as previous works have done, but now you have the opportunity to look across the entire brain. And is there any pattern coming up as to where do these plaques are formed? And a follow-up question on that, can you identify changes in diameter of the vessels nearby?
Right. So yes, these are the things that we are doing as we speak. And what I learned is that computational part is at least as important as the biology, and so it became very clear that we can only do these things if we team up with some really good computational people, and you know, the I mean, I tried many things, but something that sometimes biologists do, and it's clear that it's a big mistake, is that you know, that we use computational people as kind of a service people, no? And you know, and that never works. When things are complex. And you know, my friend Joachim Buhmann, who is the chair of computer sciences at ETH, he once was very frustrated and came to me and told me, you know, Adriano, you biologists, you think that we are the guys who calculate the p-values for you. And you know, and this never works. I mean, because they have their own ambition and their own ideas of how to advance research. And so it took me a long time to find a good way to actually to find somebody who was really interested in this, and who we could talk to as partner at equal footing. And that was really what happened with the Stanford group, with Jin, and this has become now incredibly successful. And they are actually asking all the questions that you have raised, but we are at the beginning.
Okay, so I'll wait patiently. A beautiful piece of data. Yes, thank you.
Danny?
You know, again, great lecture as always. I have a quick question regarding the first part that you show very nicely, the link between Taiwan to the ABP to the 6-3-CL1, of course, probably correlate with inflammation. But I was wondering, because amyloid, at least in the mice and the human, tend to be kind of more allocate more kind of for more cortex kind of appearance. And I was wondering if you look also on the cerebellum, and if there is kind of also correlation between the Taiwan or the ABP, or there's something that make it more affiliated more in the cortex.
Yeah, I mean, this particular fragment of the Thywan promoter that was used to generate this mice is very weakly active in the cerebellum, and this is one of the reasons why we have focused on the telencephalon.
Okay. Great, thanks.
Adriano, I translate. We're hearing the news about some business, something in the news. I don't know from where it's coming, but okay. Anyone else has a question? Scanning the people. No?
Right, I just want to say that it has been really a pleasure and an honor to have the opportunity to present our recent work to you. And again, I mean, for me, the most important thing about these type of seminars is to get to know each other, to talk science together. And if there is anything where the technologies or the reagents or the mice that we have made might be of any use to you, don't hesitate. We heed the open house policy.
Thank you very much, Adriano. The pleasure was all ours. It was really a fantastic talk. Thank you very much.
Thank you so much. Bye-bye.